Hey! Today we're going to talk about supergerms. What is antibiotic resistance? A microbe like bacteria or fungi can resist antibiotics meant to kill them. This means they are less affected by it and can grow and reproduce. Resistance is usually developed through mutation or contact with other bacteria. Mechanisms by which bacteria are able to resist antibiotics. They can limit its access into the cell by having structural defenses like peptidoglycan walls and gram-negative bacteria. They can develop pumps that expel foreign molecules or specific ones like harmful antibiotics, which in turn prevent them from lysing the cell or important genetic material. They can develop proteins or enzymes that lyse antibiotics before they can attach to their target site. Or, those proteins can interfere with binding sites by attaching to them before the drug, and changing its shape so it no longer fits. Antibiotic resistance. Let's look at some recent facts. 236.4 million cases of oral antibiotics were prescribed in 2022. This means there were 709 over-the-counter prescriptions per 1,000 persons. The wrong combination or misuse of these antibiotics drives resistance. As they are exposed to, and not killed by, multiple antibiotics, they can become resistant to them, leading to prolonged illness and stronger infections that spread from person to person. Genes can be transferred to other bacteria in a few ways. Plasmids are neatly packaged circular DNA. Transposons are sections of those plasmids that actually get transferred. And phages, which are viruses that pick up and carry DNA from one host to the next after infection. The random development of helpful mutations is usually through the transfer of DNA during division or contact. More specifically, conjugation transfers DNA that can be replicated later, some of which might be a resistance gene. This method can take some time, so cell density matters as a way to maximize contact and transport of genes. Of material through a viral carrier. This happens when a phage accidentally picks up DNA after infection, and carries it to another host cell after replication. This can take place when cells are spread out because the carrier is transferring the DNA. Relies heavily on cell concentration because it requires material to be absorbed through contact in the environment and not with a viral carrier or pilus to transport it. One of the most prevalent antibiotic-resistant bacteria is Escherichia coli. It is a 500 to 2000 nanometer in length gram-negative bacteria. It causes intestine infections and avoids antibiotics via a mutated colistin resistance gene that coats its peptidoglycan wall in a protein that makes it hard for colistin to bind to it. 100 nanometer gram-positive bacteria that is known to cause skin infections. It has an identifiable strain called MRSA, methicillin-resistant Staphylococcus aureus, which produces an altered penicillin-binding protein that has a lower affinity to antibiotics than normal. Pseudomonas aeruginosa is a gram-negative rod-shaped bacteria averaging 500–1000 nanometers in length. It is a healthcare-associated pathogen that is known for its strong resistance to antibiotics. It developed this resistance over time and contact with antibiotics, allowing its pumps to expel various important antibiotics like fluoroquinolones, beta-lactams, chloramphenicol, and trimethoprim. Klebsiella pneumoniae is a gram-negative 600–6000 nanometer long bacteria that causes UTIs, pneumonia, and bloodstream infections. It has become resistant to antibiotics by producing an enzyme called carbapamase that breaks down beta-lactam antibiotics. It causes the infection gonorrhea and has developed resistance to virtually all antibiotics. It has evolved every resistance mechanism, though mainly resists through its high genetic adaptability. It is susceptible to selective pressures throughout its lifetime, passing on its plasmid-borne resistance genes through conjugation and transformation. The most common causes for these types of antibiotic resistances developing are the misuse and overuse of antibiotics. This takes place when drugs are prescribed when they are not needed, in dosages or durations too large. Failure to treat wounds properly can also prolong an infection and allow it to spread to people who come into contact with it. Bacteria that survive exposure to antibiotics without being completely killed off by it will produce stronger generations to follow. Resistance genes can spread between bacteria through physical contact. They can spread between humans. They can be transferred by touching objects, coughing and sneezing, and a lack of proper sanitation. They will get stronger as they are passed from host to host, making the infection gradually harder to treat. Finally, we have some solutions to our problem. Hospital programs that emphasize minimal contact between bacterial strains and increased attention to sanitation. Emphasize sanitation especially in water systems and hygiene where medical infrastructure is weak would help reduce exposure to antibiotics unrelated to treatment.